All right, this is the video for chapter 3, the molecules of life. Um, and the molecules of life all have something in common. They're all made up mainly of carbon. So we call those organic compounds. They always contain carbon. Those carbons are either in chain strings or they're in rings. They will bond together in rings. And these compounds can be very large. And so we're going to be talking about the compounds that make up most of life here in this chapter. So they're complex molecules. They're built on a framework of that carbon atoms. And we're going to be talking about 4 different types of molecules of life. Carbohydrates are so important. Remember from the first lecture, I told you that sugars are going to be the key to just about everything that we're going to be talking about in biology because they are so important. So a sugar is a carbohydrate. Most of the time when we think about carbohydrates, though, it is sugars that have been put together to form much larger molecules, such as starch, that plants make. Lipids, oils, and fats are extremely important in life. You have to have these in your diet because every cell in your body is surrounded with a lipid bilayer. So every one of your over 34 trillion cells has this lipid bilayer layer. And every time you make a new cell, you have to have this lipid bilayer. So we do need lipids in our diet. But also lipids in our diets are great because what are lipids made out of? It's typically sugars anyway. It's sugars that have been arranged in a certain way to form fat. So when we eat too much sugar at one time, what's our body going to do? It's going to form it into fat molecules, and so we can break those down later for energy. Proteins, we're going to see, is all important in life too. Proteins makes up a lot of the structures of the body, but all of our enzymes, our metabolic activity is run and controlled by proteins. And then nucleic acids, our DNA, RNA, ATP, which we use, that's our energy unit, which we'll talk about later. But carbon truly is the stuff of life. They can be assembled and remodeled into many organic compounds. They can bond with 1, 2, or 3, or 4 different atoms. They can form polar or nonpolar bonds, and they can form chains or rings. Um, how do we give these on paper? How do we show the structure of these? So this is a structural model of organic molecule. This is actually glucose. This is sugar. This is the sugar that plants make from sunlight and water and carbon dioxide. So you can see that here is the string of carbons, but this forms also a ring. So we have carbons, hydrogens, and oxygens. C6H12O6 is glucose. But this is one way we draw it out. Here is the space filling model. I'm not going to ask you these models just so you can see how we show these. This is the space filling model of hemoglobin in your blood, which carries oxygen. You really can't tell much with this. This is a surface model, which you can see more. You can see the folds, and these little red areas here are the active sites in your hemoglobin. There's only one thing that's going to fit in there well, and that's oxygen. And so this molecule carries oxygen. This is the ribbon model, which shows us the different types of proteins that come together to form this, and there's our active sites in there. So from structure to function, the function of these organic molecules really begins with their structure. The building blocks of carbohydrates, lipids, proteins, and nucleic acids bond together in these different arrangements to form different types of things. And we're going to go through these as we go through this chapter. But any process in which a molecule changes is called a reaction. So metabolism, what is that? Activities by which cells acquire and use energy to construct, rearrange, or split organic molecules. So where are you going to get all your energy from in your body? From splitting sugars. So that is part of your metabolism, is splitting apart that organic molecule to get energy out of it. The metabolism allows cells to live, grow, and reproduce. It requires enzymes. Here's our proteins that increase the speed of reactions. We'll talk a lot about that a little bit later. Many organic molecules are large molecules built from smaller building blocks. A monomer is one of those building blocks. A monomer is just a single molecule. Glucose is a monomer of the carbohydrate. So it's a saccharide we're going to talk about in a minute, but it's the building block. And as we add glucose upon glucose and different sugars together, we can form different compounds, different polymers. Polymers are many of these monomer subunits put together. And we're going to see that as we build proteins. What is the monomer of a protein? It is an amino acid. But when you start putting amino acids together, it forms the polymer, a peptide chain, which becomes a protein. So, what do cells do with organic compounds? There's condensation, where you can take two small molecules, add them together to form a big molecule, and it produces water. So water forms as a product of that. Hydrolysis is the exact opposite, where we take a large organic compound, break it in half, but to do that, it absorbs water because it needs the hydrogen and the OH to attach to it as it comes apart. So water is split in hydrolysis. Water forms in condensation. So here's condensation. We have two small molecules that come together and they have an OH on the end, and so they are joined together with the oxygen, but water is formed. And here's hydrolysis. It's the exact opposite. We have the large molecule, water is added to break them apart. So that is a big part of our metabolic activities. We're either putting together. So as we move to the next test, we're going to spend a whole chapter on putting together that plants are able to put together these molecules through the energy of the sun to form sugars. But as we break apart sugars to get energy, we are taking that large molecule and breaking it apart, and it will form water. So that's why these are important to talk about. Functional groups, we're not going to spend much time on this. Be thankful for that. I know some of the classes, they make you memorize these functional groups. But a hydrocarbon is an organic molecule that consists only of hydrogen and carbon atoms. But typically what happens is most biological molecules have at least one functional group added onto it. It is a grouping of atoms that come together, and it forms, it's attached to the carbon chain, and it gives that molecule a certain property, such as polarity or acidity, or will it dissolve in water as a functional group? Will it not dissolve in water is another functional group. So here is a list of the functional groups. Again, I'm not going to make you memorize these. Just know that there are functional groups attached to these molecules of life that give them their unique properties. So, we've got all kinds of different functional groups here. So, what is the chemical reaction? When molecules interact with each other, they can exchange atoms. And arrows show the direction and the reaction of the products. Some important organic molecules. All living things are made up mostly of carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. Huge numbers of arrangements. So let's look at this first group, the carbohydrates. They're the most plentiful biological molecule in the biosphere. So that might be a good question. What is the most plentiful of the biological molecules of life on Earth? It is the carbohydrates. We're going to see here just in a minute that trees, a lot of weight in trees is made up of these sugars just put together to form these large molecules which form a solid structure. Then the plant just pumps an enzyme, a protein in there called lignin that actually forms it into woody tissue. So, but most of what we see out in plants that gives it its weight is made up of sugars. And so Again, sugars are all important. Cells use some carbohydrates for structural materials. Others are stored for instant energy, stored or for instant energy. And we're going to see that as we go through these. So what is a carbohydrate? It is carbon, hydrogen, oxygen, usually in a 1-to-1 ratio. So one part carbon, and then twice as much hydrogen, and then the same amount of oxygen. 3 types of carbohydrates in living system. Monosaccharide. That word saccharide, focus on that part. Saccharide is Latin for sugar. So it is sweet. So a monosaccharide is a single sugar molecule. Most plentiful is glucose, made from plants in photosynthesis. Fructose, which is made from plants for fruits. An oligosaccharide is a small molecule. So this is the monomer, monosaccharides. So if you put 2 of them together or 3 of them together, you get an oligosaccharide. The best example of this is sucrose, which is 1 glucose and 1 fructose put together. And when you do that, you take 2 monosaccharides and it forms an oligosaccharide, sucrose, and you all know sucrose as table sugar. It's it's the most common sugar on the planet, the most common oligosaccharide. And then a polysaccharide is when you take these monomers of, we're talking about sugars here. You take 1 sugar, put it together with another, you form an oligosaccharide, then you start adding sugars and sugars and sugars and sugars to make a giant molecule. That is a polysaccharide. Poly is Latin for many. So 1 to many. So those are how carbohydrates are built in nature. So a monosaccharide, 1 sugar unit, is the simplest carbohydrate. It's used as an energy source. It's instant energy. So here's glucose. This is our main source for energy. Or it can be used for structural material. When you put these together and start building large chains, it can be used for structural material. The backbone of these sugars is 5 or 6 carbons. Glucose has 6 carbons. And so it is an organic compound. It's a carbon chain. And here is just some different ways to draw this out. Since it does form into a ring, you can just draw just that ring. Each of those blocks are carbon until there's the oxygen. Then you can add the different things to it. But here is the chemical formula for glucose, C6H12O6. Now, an oligosaccharide, very short chains of monosaccharides, usually only 2 or 3. So here's sucrose, a glucose and a fructose put together. So what's it called when we take 2 small molecules and we put them together? That is called condensation, remember? And so that forms sucrose with water. So remember, condensation, water is a product of that. Polysaccharides, when you start putting these monosaccharides together, they can form chains, they can form spirals, or they can be arranged into a lattice work. We'll see. The most common polysaccharides is cellulose, starch, and glycogen. All of those consist of glucose monomers. Each has a different pattern of covalent bonding and different chemical properties. So let's take a look at these. Here is cellulose. Notice what each of these is a glucose molecule. So what happens is, if there's a really nice sunny day outside, the plant, like today, the plant is out there, it's got its leaves out, there's plenty of moisture in the soil, there's plenty of water. So he's going to take carbon dioxide from the air, water from the roots up through, and with the energy of sunlight, and he's going to start making glucose molecules. Now, he's going to make a lot of glucose molecules on a day like this because it's so sunny and conditions are just right. He's going to make too much. So what happens when he makes too much? Well, he can start to put those together, and that'll be for energy. Like if tomorrow's going to be a rainy, stormy day, there's not hardly any sun, the plant's not going to be able to make it. Or in the middle of the night, if the plant needs energy to, which it does constantly, it can take these glucose molecules. This is starch here. And so plants put the glucose molecules together and it forms these coils. This is starch, and it uses it for energy reserve. So energy will be reserved in as starch. Now, the plant can also take these glucose molecules, arrange them here in a lattice work, and this forms a tougher fiber, and this is what is called cellulose. We can't digest cellulose. We can digest starch. We get a lot of the sugars that we need by eating starches and carbohydrates. Where do we find carbohydrates in our diet? Mainly in breads, in any wheat, any grains. It's full of carbohydrates. Potatoes, a very rich form of starch. Pastas, which is made out of flour. And so we can take those and break off the sugar units. That's what our digestive system does. As we digest these, we break off each of these individually so that we convert what we eat into sugar. Then we can store that, or we can use it instantly as sugar. But for cellulose, we can't digest that. We can't break these off through our digestive system. But we still need this as part of our diet. You all know what it's called in our diet when we have cellulose. This is dietary fiber. We can't digest it, but it helps things keep moving through the digestive system. And then last but not least over here is glycogen. This is what we form. Remember when plants have excess sugar, they store that as starch. They start putting glucose. Makes a big giant polysaccharide called starch. When we eat too much sugar, let's say, for lunch today, you're in a rush, so you grab 2 Snicker bars and a Mountain Dew and you down them real quick. Now you got all this sugar in your body. Too much sugar in your bloodstream at one time is bad. So your body is going to release an enzyme that's going to take that sugar molecule and start putting them together. And it's going to put them together like this in branches. So each of these is a glucose molecule. And this is called glycogen. Now what your body's going to do with glycogen after that is it's going to form it into tails, put it on a glycerol head, and form a fat. And so, so this is how we store energy in animals, is through glycogen. So here is cellulose. You see the nice pattern. And so this forms a very rigid structure. This allows plants to grow up and so that they can stand tall for a while. Once they get too tall, cellulose is not enough to keep them, and they'll break and fall over like a tomato plant. Once it gets to tomatoes, many times they'll break and fall over. So what plants do then, if they're going to be annual plants, if they're going to survive more than 1 year, they'll start pumping enzymes in here, which will form this cellulose into a much tougher substance that is very fibrous, and we call it wood. And so plants can take the sugar and make cellulose, which gives plants the ability to grow up, and also wood, which allows them to grow really tall. Here is starch as it comes and coils up. Each of these is a glucose molecule, so this is for an energy reserve. And here is glycogen. When we eat too much, we store their sugars as this, and this will be turned into a fat. So this will be used in the head of a fat with some fatty acid tails attached to it. So that is carbohydrates. Now the second organic compound we're going to look at is lipids. So greasy, oily, they must be lipids. Lipids function as the body's major energy reserve. Remember, when we have too much sugar, we store it as glycogen. We turn that into a fat. And so that is our energy reserve. So that it's not that important for us because we have so much food available. But I keep using wolves as an example. The wolf may get a kill today, and what they'll do is they will gorge themselves on that kill. Because it might be a week or 2 weeks before they're able to get that energy back into them again. So what are lipids? Fatty, oily, waxy, organic compounds that are insoluble in water. Most lipids incorporate fatty acids. They're simple organic compounds of a carboxyl group joined to the backbone of 4 to 36 carbon atoms. This forms a long chain of carbon atoms. But you need to know the difference between these two. Saturated fats use only single covalent bonds so that the tails on the fat, usually 2 or 3 tails, they're straight. And so they form little blocks and they're able to pack together really tightly. So at room temperature, fats are solid. Now, oils, on the other hand, are unsaturated. They have at least one double covalent bond, usually more, and that causes those tails to kink out. And when the tails kink out, it doesn't allow them to pack as tightly, so they do not form those saturated fats. So saturated fats, animal fats, they only have single valent bonds. They pack tightly. Unsaturated, they have one or more double covalent bonds, and they're kinked. And so here is a saturated fatty acid tail. You see it's only single covalent bonds, forms a straight tail. Here is another one. See, there's a double covalent bond. There's a double covalent bond, which causes the tail to kink out so that they can't pack real tightly together. And here's one that has 3, and so it's even more kinked out. And so when this happens, they become unsaturated. They can't pack in together, so they stay usually liquid. So oils remain liquid at room temperature. So fats are lipids with 1, 2, or 3 fatty acid tails attached to a glycerol. Triglycerides, this is the most abundant lipid on the planet. They're neutral fats with 3 fatty acid tails attached to a glycerol head. They're the most abundant energy source in vertebrates. We store these triglycerides. This is our energy source. Concentrated in adipose tissue for insulation and cushioning. Adipose tissue is just a connective tissue. It looks like a honeycomb type thing. And so what your body does is it stores the fat in those. So it knows where it is when it needs energy. It can go and break it apart. But also it stores it in that to add a layer of cushioning. And so it provides the softer parts of our body. So here is triglycerides. Here it is at the 3 fatty acid tail. All right, phospholipids. Now these are important. Phospholipids, they have a polar head containing a phosphate group and a non-polar fatty acid tails. The heads are hydrophilic. Hydro means water. Philic means love. That's from philios, the Greek word for brotherly love. Philadelphia is the city of brotherly love. So they are water-loving. The tails, though, are hydrophobic. Phobic means afraid of or repulsed by. And so the tails do not come in with water. So the most abundant lipid in cell membranes. These phospholipids is what forms that layer around each and every one of our cells. That is why our cells are able to determine what comes in and what goes out, because water will not pass through it because the tails are hydrophobic. And so your cell then will allow things to be exchanged back and forth as it goes through that membrane. So here is our lipid bilayer. So if this is the outside of the cell, things cannot pass through this. So now the cell, what's going to happen is you're going to have all these different types of proteins sticking up through here. That when you need something inside your cell, it's either going to grab it and pull it in, or it'll form a channel where only that thing will naturally flow in. And so these phospholipids are very important to life. Waxes is also a lipid. Its long fatty acid tails usually attached to an alcohol or a carbon ring. It's used for protection. It's water repellent. Plants make wax to cover their leaves so that water is not evaporated out, because it has to keep that water in there to make sugar. So wax will keep that from evaporating out. Remember, because the tail of the fat is hydrophobic, and so the tail is against the outside layer of the leaf so that water will not pass through it as it evaporates. Steroids is another lipid. It's basically, it has no fatty acid tails. It's just the head. Cholesterol is probably the most common of the steroids. And so our bodies use cholesterol to be remodeled into bile salts or vitamin D, which we need for bone structure. And we are learning more about vitamin D here. Vitamin D is actually helps us to be immune to many viruses. And so part of the reason why the flu doesn't happen in the summer is there's not as much sunlight and people aren't out in the sun as much and our bodies don't have as much vitamin D. That's some things that we're starting to learn about our immune system. But that comes from a steroid in our body, cholesterol. All right, so that is the lipids. Now what about, so we've gone through carbohydrates and lipids. Now let's talk about proteins. Proteins are all important in our bodies. They're the most diverse biological molecule. We use them for structural things. Your hair is made out of protein. We use it for, we can break apart proteins and get energy from those too. We can convert those proteins into sugars, and actually we put it into, we're not there yet, but we can put it into the process of where we break down sugars and form ATP. There's defense proteins that help us to stay healthy. They can recognize outside invaders. But cells build thousands of different proteins by stringing together amino acids in different orders. So amino acids are the monomers of proteins. Know that for the test. What's the monomer of a carbohydrate? It's a sugar, glucose usually, fructose. What's the monomer of a fat? Well, and most of the time it is sugar also. It's put together to form those long structures. But what's the monomer of a protein? It is amino acids. So protein, there are polymers of amino acids. There's 20 different types of amino acids in nature. 9 of them are absolutely essential for us to have in our diet all the time to make the proteins that our body needs. There's different levels of protein structure. Now, why is this important? Know on the test that this is the compound where structure equals function. For proteins, structure equals function. Protein is shaped in such a way that it will do only one thing. And so as long as it keeps that shape, it can do the function that it was made for. So there's different types of structure. The primary structure is just the sequence of amino acids. That's what your DNA codes for. So when your DNA unzips, it gives you the code for this amino acid goes here, this amino acid goes here, this amino acid, this amino acids, and these proteins can be hundreds or thousands of amino acids long. So that's the primary structure, the order of the amino acids from the first to the last that makes the protein. The secondary structure, as these amino acids start to be put on, it starts to make this long chain, it's going to start to fold it. It can coil, it can fold together like sheets, and so there's hydrogen bonds that keep them into this secondary shape. The tertiary structure is when that it comes all together, so it's into coils, but then it's added together into a large molecule that gives it a specific shape. And so it forms the functional protein. It's the active site of the protein. Now, quaternary structure is when, for a lot of our enzymes, we need more than one protein. So the protein is made and folded, and then another protein is made and folded, and then they're combined together to form a larger one. So that is this fourth type of structure. An example of that is hemoglobin. We've already looked at that a little bit. That's what carries our oxygen in our blood. So polypeptide, I hate that your book does this. Poly just means many. Peptide means a chain of amino acids. It's protein. So it'd been much easier just to say this is protein formation. So here is some amino acids put together, some more amino acids added together, and so there's the 4 of them, just the names of them out there. And so that is the primary structure of a protein. The secondary structure, it can twist into a coil. It can be a sheet held together by hydrogen bonds, but it's the same pattern secondary structure occurring in many different proteins. So it's the same pattern, but the amino acids are going to be a little bit different. All right, so there's the primary structure, there's the secondary structure, and there's the tertiary structure as the coils and the sheets fold into a functional domain, such as a barrel or a pocket. In this example, the coils of a globulin chain form a pocket. So there is a pocket there inside of those coils that something will fit into. And it's usually only one thing will fit into that very well. And there is our quaternary structure where there's more than one protein come together to form the actual protein that's going to be used for a function. So here's your hair. Many proteins aggregate by the thousands into much larger structures, such as the keratin fibers that make up our hair. So those are made up of proteins. So we can use proteins structurally. We can use it for metabolic activities. We use it for activation for these reactions that take place in enzymes. So protein structure is a peptide bond. So all that means is, what is a protein? It is amino acids attached together. Then the secondary structure is they coil. The tertiary structure is they are folded. So that's just a review of what we just talked about. This again is another review. There is our primary structure, just the order of amino acids. The secondary structure, coiled or folded. And then the tertiary, where even that is folded to make the active site. So why is protein structure so important? They will function only as long as they maintain the correct shape. Changes in the protein shape has drastic consequences to our health. So we want to make sure that our proteins stay the correct shape. What is it that can cause a protein to lose its shape? Excess heat will do that. If you take an egg and crack it, the white of the egg is protein. So eggs are high in protein. But as you add heat to it, you'll notice that clear white turns, you can't see through it anymore. That is showing that those proteins have been unfolded and they've got a new shape, so they're no longer see-through. Now it doesn't function the way it's supposed to function. Now it's still amino acids that we can eat and digest so that we can make our proteins. So when we eat proteins, we digest the protein, pull apart all the amino acids, so that now our body has the amino acids we can grab and put together to form new proteins. But as you add heat to protein, it unfolds and it loses its function. That's why whenever we get overheated, that you start to get lightheaded, you can't think, you can pass out from heat stroke, because your enzymes start to unfold and your metabolism can't function the way it's supposed to. When you're out in the cold and your body temperature starts to drop, so getting colder causes these proteins to start to unfold. And as they start to unfold, they can't carry what is needed to break down sugar, so you start to lose energy. Your mind doesn't function the way it's supposed to. You start to stumble until you finally fall down. And so that is what happens in our bodies when our proteins start to lose their shape. So a protein that loses its shape, we call it denatured. So they function only as long as they can maintain their correct three-dimensional shape. Heat changes in pH. I was telling you, heat can change that. pH changes in your body can unfold proteins. That's why your body is going to fight to keep pH around 7. Salts in the body. Detergents can disrupt the hydrogen bonds that maintain a protein shape. Now, this is critical right now. The soap. Why is it that during this pandemic they're telling us to wash our hands, wash your hands, wash your hands? Because the virus that we're dealing with here is made up of basically just 3 things. There's an RNA, which we're going to get to in a minute. It's a nucleic acid. Then it's got a protein coat around it. So it's made up of protein. That protein is holding that RNA so that it can invade your bodies and take over. Also, this particular virus also has a plasma membrane around it that it is taken from the last person it was in. And so it has this fat layer around it. Now, soap will do several things. Soap will actually break apart the fats. And so as you wash your hands, you're actually breaking apart that envelope around the virus. They call that an envelope. It's a plasma membrane. And so you're breaking that apart. Now, why do you use hot water? Because heat can start to unfold the protein, and also the soap will help to unfold that protein. Once that protein is unfolded, that RNA is released, and it can no longer invade your body. That's why washing your hands with hot water and soap is so critical here, because we're breaking apart the protein and we're breaking apart the lipid membrane. Now, when a protein loses its shape, it can no longer function, so it's called denatured. So examples of proteins, we have enzymes. Enzymes are transporters. We do have transporters such as hemoglobin. Enzymes are catalysts. They get our chemical reactions going. Without enzymes, we wouldn't be able to break apart sugars. So they are all important too. Transporters, hemoglobin, move things throughout our body. Oxygen, we have some sugars. It can be structures, like we talked about hair. It can be fibers in our body that is made up of proteins. Hormones, such as insulin. Insulin is all critical. If you've got too much sugar in your blood, that's a bad thing. And so your body produces insulin. Now look, insulin is a 51 chain of amino acids. Now look at the chances of just that happening by chance. So you got too much sugar in your body, you're going to die. You will quickly go into ketoacidosis. Oh no, what's going to happen? Well, your brain says, well, let's make some insulin. So what's the chances of making insulin just if it just starts making things? There's hundreds and there's thousands. There's millions, there's billions, there's trillions, and then quadrillions. That's an amazing amount of molecules that would have to be made before you made even one insulin. But guess what? Your body will make it every single time. How can your body make it insulin perfect every single time? Because part of your DNA is the code for what amino acids, and 51 of them go. This one goes here, goes here, goes here, all the way down the line for 51. So when your body needs insulin made, it unzips that portion of DNA, it makes a copy, it takes it out to the ribosome where this protein is made, and so but it'll make it perfectly every time. This is just mind-boggling for the first time that insulin was made, and the importance of it was realized somehow. All right, so what is a prion? A prion is a protein that has been misfolded. This can be disastrous in the human body. When our proteins misfold, it will cause disease. Mad cow disease, bovine spongiformis encephalitis, is a misfolding of the proteins in the brain. It causes holes in the brain tissue. Creutzfeldt-Jakob disease in humans is always terminal. It's lethal. It is a misfolding of the proteins in the body, especially the brain, that it just will not function right now. Scrapie in sheep is prion disease. All are infectious diseases characterized by deterioration of mental and physical abilities that eventually causes death. So you can see that the proteins folded correctly is critical. When you get these diseases that start to misfold the proteins, disaster comes about. So here is an example of brain tissue with the variant Creutzfeldt-Jakob disease, and it just causes these misfoldings all over the place so that they can't function as brain tissue. All right, so that is proteins. We have one organic molecule left to go, nucleic acids. Now, what are nucleic acids? They function to store and transfer information. It's a blueprint. And that information is what amino acids go where to make the proteins. This is actually nucleic acids. It's amazing. It is a language. And the language is inside of our cells. So what is a nucleic acid? It's composed of polymers of nucleotides. So what's the monomer of nucleic acids? Nucleotides. What's the monomer of a protein? Amino acids. What's the monomer of a carbohydrate? Nucleic acid, ATP. ATP is the energy source in our body. Now, when we plug into the outlet on the wall, we think about amps and volts carrying the energy through the appliance that we need. This molecule carries the energy through a phosphorus bond. So this is adenosine triphosphate. It started off with two phosphate groups, but when you add a third one to it, it becomes triphosphate. It has now more energy, and so now your body can break off one of those phosphates, get that energy, and use it for a metabolic purpose. So this is important in energy transfer. So there's a nucleic acid. It has this backbone here, and then the nitrogen bases attached to it. So a nucleic acid is polymers of nucleotides in which the sugar of one nucleotide is attached to the phosphate group of the next, and so on and so on. RNA and DNA are nucleic acids. So this is the guys that hold the information about how your body is going to make all the different proteins that it needs to make. So RNA contains 4 types of nucleotide monomers, including ATP. It's important in protein synthesis. Your DNA holds the codes, but your proteins are made outside of the nucleus at the ribosome. So how are you going to get that information from the DNA out to where the protein's made? Well, RNA comes inside the nucleus. The DNA is unzipped. The RNA will then come and make an exact copy of that portion of the DNA, then carry it out to the ribosome, and then they will put together the amino acids there at the ribosome. So that it is critical in protein synthesis. And another important, we already talked about ATP, that is our energy unit. Remember that? It functions as our energy carrier due to the 2 high energy bonds. And here is our DNA, deoxyribonucleic acid. It is 2 chains linked together and then coiled. It's a double helix. It contains all the inherited information necessary to build an organism, coded in the order of nucleotide bases. So it is a blueprint. It is a complex language. Here are nucleotides of DNA. You have adenine, guanine, cytosine, and thymine. The adenine will always bond to thymine. So A will always bond to T. So it's A, T, T, A. Guanine and cytosine always bonds together, G, C, C, G. And so as they go together, they form this double helix. So it uses the order of those bondings to tell your ribosome what amino acid goes where. So here is the nucleic acids here. So here's our A and our T, our C and our G. And so this makes a language. You'll realize that when we put a man on the moon, we did it with binary code, which is only dot dash. That's what computers used back in that day, was just that binary code. This is actually a quaternary code. It has 4 different types, but as they come together, it forms a language. This language is complex, and it is plentiful. Oxford did this not too long ago, Richard Dawkins and his group, they wanted to determine how much information was just in one cell of your body. So you have 34 trillion cells. Every single cell in your body has a complete DNA. So every cell in your body has the code on how to make all of you. How much information is in each of your cells? Well, they asked this question. If you would take this AT, so you start typing this out on a regular piece of paper, 12 font times New Roman. Type it out. Just one cell. How many pages do you think you would fill up with just the code out of one of your teeny little cells? Most people, when I ask that in class, well, they'll say 10, 100, 150. Actually, and there's a video online that you can go and watch this that they made out of Oxford. If you took the papers of what you typed and started stacking them, the stack of papers would be over 360 feet tall. Dawkins said that that is more than having 60 complete volumes of Encyclopedia Britannica stacked together. There's that much information in each and every one of your cells. That is an amazing amount of information that's in your cells. And it's not just random information, it is precise information. It is a language, and it tells your body how to make all the proteins that you need to live. As we go through photosynthesis and respiration and metabolism, we're going to see these proteins are absolutely critical for life. So protein is needed to build DNA. DNA is needed as the information to build proteins. Proteins are needed to make ATP. ATP is needed to make proteins. So which came first, the chicken or the egg? That's the question here. They all had to be there at the exact same time in order for this to function properly. How does that happen? That's something you can just think about. This is just a great slide on your PowerPoint that you can use to study with. It gives you all four of the organic molecules, carbohydrates, lipids, proteins, and nucleic acids, give you the basic information about each. So that is the end of chapter 3. We only have one more chapter to go before our first test. Chapter 4, when we're going to start talking about the cell. I love chapter 4. To me, it's just so interesting to see the complexities inside of every one of our cells. We already looked at, just here, how much information is in the DNA in each of your cells. An astounding amount of information. And what your cell does is just as amazing. Every single cell in your body is like its own little city. And it has all the things that a city needs in order to function and survive. So that'll be next chapter. I will put that on your Moodle here presently. So thanks for watching. And just to let you know, every now and then I'm going to throw a quiz into your Moodle week. And it will be information that you can get from watching these videos. So it's just a way to make sure that you're keeping up with the videos and trying to keep up with the book. As I've told you before, this class is going to start going, and it's not going to slow down. You've got to keep up. So keep up with your reading, keep up with your video watching, and we will be testing here soon. So thanks. We'll see you soon.